So, for several different reasons, molecules of substances are in constant motion. The relative motion differs based upon the phase of matter, gas, liquid, or solid. As molecules move, they collide into each other and move in different directions. The effect of these collisions will be influenced by the size or mass of the molecule. As a result of this motion and resulting collisions, in an open environment, molecules tend to move from an area of higher concentration to an area of lower concentration. This is known as free diffusion. This diffusion occurs until the substance has dispersed equally throughout the environment. Though the molecules or ions continue to move, there is no net movement because the substances are equally dispersed. This is called equilibrium. So once again, in an open environment, molecules or ions tend to move from a place of higher concentration to a place of lower concentration. This is known as free diffusion. Molecules tend to spread throughout its environment until equilibrium is reached. And equilibrium means that the molecules are now evenly distributed throughout the environment and diffusion can no longer occur. The plasma membrane serves as a barrier to free diffusion. You now know that the unique properties of the plasma membrane may pose difficulty for some substances to enter the cell. We refer to the plasma membrane as being semi- or selectively permeable because it can indeed allow some substances to enter or leave the cell and exclude others. For substances to freely enter the cell by diffusion, they must be able to be dissolved in lipids because of the lipid tail of the phospholipid bilayer. They must be small enough to pass through static protein channels or somehow be assisted by a protein carrier molecule. The animation shows two cells. One is impermeable to a particular molecule and one is not. A substance, represented by the dots, is able to diffuse into the cell on the right, but cannot enter the cell on the left. In essence, the plasma membrane is serving as a barrier to diffusion. This is an important specialization that contributes to a cell's ability to maintain a distinct and different internal environment as compared to its external environment. It is important to note that a cell's selective permeability is often episodic rather than constant, meaning that given varying conditions, a cell may be able to change its permeability, allowing substances to enter or leave in one instance and then not allowing the transfer in another. It should also be noted that some substances easily diffuse through the plasma membrane. They include, but are not limited to, oxygen, carbon dioxide, and lipid-soluble vitamins. There are other substances that only diffuse across the plasma membrane with help. Facilitated diffusion requires a transmembrane protein to help carry substances across the membrane. This type of facilitated diffusion is required for substances like glucose that are too large to pass through typical membrane channels. In facilitated diffusion, substances bind to the protein. The binding causes the protein to change its shape, opening a channel to the inside of the cell. It is important to note that this process is still diffusion and is still passive, and therefore the substances move down its concentration gradient from a higher concentration to a lower concentration. Sometimes transmembrane proteins form channels that shield substances from the anatomy of the plasma membrane. Water readily diffuses in and out of a cell because of protein channels known as aquaporins. Osmosis is the diffusion of water across a membrane. We need to be clear that when we refer to diffusion, we are not referring to the amount of water on either side of a membrane. Concentration is more a product of the differences in how close the water molecules are to one another. If molecules are further apart from one another, then the concentration is less. The left side and the right side of the screen depict an equal volume of water. When a solute is added to the right side of the screen, the volume remains the same. However, the solutes push the water molecules further apart from one another. The concentration of water molecules has now decreased as compared to the left. So to be clear, osmosis is the diffusion of water across a membrane. Osmosis occurs when the concentration of water differs on either side of the membrane. In osmosis, the concentration of water is determined by the concentration of non-permeable solutes in the water. And finally, as the concentration of non-permeable solutes increases, the concentration of water decreases. So here is an example. The left side of the depicted U-tube is filled with water. The left and right side is separated with a membrane that is permeable to water. Over a period of time, the water will diffuse from the left side to the right side until equilibrium is reached. A non-permeable solute is introduced to the left side. Non-permeable means that the solute is unable to cross the membrane. The solute decreases the concentration of water on the left side. Osmosis is the diffusion of water across a membrane from a higher concentration to a lower concentration. Therefore, water moves from the right side to the left side. Tonicity is an example of how the external environment of cells can alter the flow of water. If the extracellular environment presents with a higher concentration of non-permeable solutes than the cell, that is, a higher concentration of solutes that cannot enter the cell, then the concentration of water on the outside of the cell would be less than inside the cell. Osmosis is the movement of water from higher concentration to lower concentration. In this case, there is a higher concentration of intracellular water, and water moves from the cell to the extracellular space. Because of the loss of water, the cell begins to shrivel or crenate. When a solution has a higher concentration of non-permeable solutes than in the cell, the solution is called a hypertonic solution. Conversely, if the extracellular environment presents with a lower concentration of non-permeable solutes than in the cell, water will tend to move from the extracellular environment into the cell. The cell will then swell and could eventually rupture as excessive water diffuses in. When a solution has a lower concentration of non-permeable solutes than in the cell, the solution is called hypotonic. When the cells are bathed in a solution with the same concentration of non-permeable solutes, no net diffusion takes place. There is equilibrium between the concentration of water inside and outside of the cell. When a solution has the same concentration of non-permeable solutes as in the cell, the solution is called isotonic.